Hello everyone. In this short video, I'll introduce RNAwait, our latest innovation for RNA stabilization in tissue samples. As many of you know, preserving RNA integrity after tissue collection is a major challenge. RNAwait provides a practical, reliable solution to this problem. It removes the need for immediate freezing while maintaining both RNA and tissue morphology quality. Once tissue is excised, the cellular environment is instantly disturbed. Oxygen levels drop, nutrients are lost, and stress responses begin. As a result, cells change their gene expression patterns and RNA starts to degrade. This can lead to misleading transcriptomics data and poor quality RNA for downstream analysis, such as RNA sequencing, qPCR, or digital PCR. The classical solution is formalin fixation and paraffin embedding of the tissue. Such so-called FFPE samples are the gold standard for histopathology and immunohistochemistry because they preserve morphology very well. However, the fixation process heavily fragments RNA and chemically modifies it, creating artifactual nucleotide changes. So, while FFPE samples are great for microscopic analysis, they're often unsuitable for RNA sequencing and other molecular methods. This problem is even exacerbated by non-standardized FFPE procedures, amongst others too long fixation times. And there is even continued RNA degradation over time with old tissue blocks typically of very poor quality. Another common approach is snap freezing the tissue in liquid nitrogen. This preserves RNA very well if done immediately after excision. But freezing requires access to specialized equipment and cold chain logistics, which isn't always feasible in the field or in operating rooms. In addition, once such frozen samples thaw, degradation resumes extremely rapidly. RNAwait is Inactive Blue's newest reagent designed specifically for tissue collection and RNA stabilization. It rapidly permeates fresh tissue, stopping both degradation and stress-induced transcriptional changes. The tissue can simply be submerged in RNAwait at room temperature. No freezing or immediate processing is required. This makes RNAwait extremely convenient for fieldwork, clinical settings, or multi-site studies. In just 4 simple steps, the tissue is preserved and RNA extracted. Step 1. Cut the tissue so that the maximum width is 5 mm in one direction. 2. Incubate the tissue in RNAwait for at least overnight in a fridge. 3. Take the tissue out of the container and store it in a new container at room temperature or in the fridge or freezer. It doesn't matter. And finally 4. Extract the RNA when it is convenient for you. Here you can witness RNAwait's performance under different storage conditions. 2 genes were quantified in 2 different murine tissues using RT-qPCR. The RNA remained stable in the tissue for 1 month at 4 degrees, 1 week at room temperature, and even 1 day at 37 degrees Celsius. All without RNA degradation. This means you can collect and store tissue anywhere and still obtain high quality RNA later when you have time. Another huge advantage of RNAwait is its robustness during freeze-thaw cycles. After an overnight incubation at 4 degrees, tissues can undergo up to 5 freeze-thaw cycles without loss of RNA quality. That flexibility makes RNAwait particularly useful for biobanking, where samples may be repeatedly retrieved for analysis. Or, if the freezer breaks down, you should not worry too much. And finally, RNAwait preserved tissues also maintain acceptable morphology. Here I show a kidney treated with RNAwait, then fixed and stained with HNE. The structure is comparable to that of a standard FFPE section, but now with intact RNA for molecular assays. And this dual compatibility, histology and RNA analysis is a major advantage over other reagents. To summarize, The main problem is RNA degradation and transcriptional drift after tissue excision. Traditional methods like FFP and freezing each have significant drawbacks. RNAwait offers a simple, flexible, and reliable alternative, preserving both RNA and morphology without the need for immediate freezing. So it's ideal for biobanking, translational research, and clinical workflows that rely on high-quality RNA. Please have a look at the RNAwait page for more information by scanning the QR code, or ask a sample for testing in your lab. You'll be amazed by the quality and the affordable price tag.